Okay, let's move on and start talking about another system other than the nervous system that is used by animals to move signals around in the body. And that's going to be the endocrine system. Now, the endocrine system is going to be a system by which cells can communicate that are distant from each other. They're not physically close to each other in an organism. And this is going to do this by not actually having the cells like neurons. Now remember, neurons are going to be cells in the nervous system that actually are in close proximity to the cells that they want to communicate with. Synaptic clefts are very, very small distances. But in the endocrine system, they can be separated by quite a distance. And instead of actually having the close proximity, the endocrine system is going to use the circulatory system to deliver signal molecules to other parts of the body or the cells that they want to communicate with. And these signal molecules, instead of, remember in the nervous system, we call the signal molecules neurotransmitters. In this case, in the endocrine system, these are going to be hormones. Okay, now hormones, once they leave the signaling cell, they're going to bind to a receptor on the target cell and cause some sort of change or communicate some sort of signal. Now, there's some contrast with the nervous system in how this is going to work. Now, the first one we've talked about already is that we're providing distant communication, and this isn't going to be achieved by some extension of a cell like an axon on a neuron. Instead, we're using the circulatory system to carry these signal molecules around the organism's body. And this is going to yield several different things. First off, this means that endocrine signals are going to be quite a bit slower than the signals transmitted by the nervous system. But also, this also has the effect that endocrine signals tend to have a longer duration, or the effect that they have tend to have a longer duration than the effect of the nervous system as well. So a couple key differences between the actions of the endocrine system and the nervous system there as well. Now, that's not to say that these are always completely separate systems. Actually, turns out that in several key ways that we're going to talk about more a little bit later, the nervous system and endocrine system tend to be fairly closely linked, and sometimes are actually referred to as the neuroendocrine system. And there are generally particular organs that are specifically called neuroendocrine organs or neuroendocrine glands or something like that. And again, this is something that we're going to come back and talk about a little bit more later, but just kind of hang on to that fact for the moment. Now, just a couple basics of the endocrine system overall. An individual endocrine gland, a gland is going to be some organ or some set of tissues that is producing hormones, and each one of these can produce many different hormones. It's not limited to a single signal or a single hormone that it can send. Also, any one hormone can be sent by several different glands. So any given hormone does not necessarily only come from one specific place in the endocrine system. So it can come from several different places. So that makes it a little bit more complicated when we're talking about the functioning of the endocrine system. But to take it even a little bit more complicated itself, any hormone can have more than one target cell. And so these hormones can be acting in different tissues at different places, in different organs, and often the functions that it has in these different tissues and different organs, in different target cells, is going to be different as well. The particular function of some hormone on your heart and on cardiac muscle can be very, very different than that exact same hormone and its function on the smooth muscle in your intestines. So this is something that we're going to find out, and you're going to have first-hand experience with as lab goes on further down this quarter. And then also, any given target cell may respond to multiple different hormones, and again, have different responses to those different hormones. So really, there's a kind of very complicated web of different glands, different hormones, where they're produced, and the types of effects that they have on these different target cells. And then to make it even a little bit more complicated, some molecules are both a neurotransmitter and a hormone. They can be used in both roles. It just depends on how are they being transmitted in any given point. A good example of this is epinephrine. So you look here, epinephrine in many parts of the nervous system is used as a neurotransmitter, but can also be dumped into the circulatory system, into the blood, carried around the body, and acted as a hormone as well. So another really overwhelming basic of the endocrine system is that hormones are going to operate at very, very low levels. And this is in pretty large contrast to neurotransmitters in the nervous system. Hormones can exert their effect at levels as low as 1 picogram. This is 10 to the minus 12th grams per milliliter. So this is really very, very, very low concentrations. They can still have physiologically relevant effects. And this is actually way, way lower than neurotransmitter levels in the synaptic cleft when the nervous system is sending a signal. Now, the opposite part of that is that hormones are being dumped into the circulatory system, so they're going everywhere, whereas in the nervous system, a synapse is a very, very small space, and so it's delivering a very targeted amount of neurotransmitter, not kind of dumping in the circulatory system and let it circulate around the entire body. So there's some contrast there. However, since they are at these very, very low levels, gets back there. This signal is often going to be amplified by a second messenger cascade. You'll probably remember these from cell biology. And one of the really big advantages to using a second messenger type messaging system to get messages across the cell membrane is that we can get ridiculous amounts of amplification of that signal. And that's exactly one of the big things that hormones are going to take advantage of. We can really amplify that signal whenever this gets to any particular target cell. And so we don't need very, very high concentrations of it. Okay, so those are some of the basics of the endocrine system there. Let's talk about some of the different kinds of hormones that can come up and be used in the endocrine system. So there's several different classes, and the first class that we're going to talk about are peptide or protein hormones. And like most proteins or most peptides, these are going to be composed of some specific amino acid sequence. And really, in the end, most animal hormones are these peptide or protein hormones. This is kind of the largest class of those. And one big example of this is one hormone that I imagine that you have heard about before, insulin. Insulin is involved very heavily in regulating metabolic substrates, particularly glucose in the blood. And there's something we're going to talk a lot more about, especially as we're talking about regulation of metabolism. But this is one of those specific protein hormones. Also, we have the amines, which are just individual modified amine molecules. And one big example of this that's used very often nearby is the catecholamines. This is particularly, again, norepinephrine and epinephrine. Again, you'll probably remember that these have been used as neurotransmitters as well. They are not only hormones, but often these are used as neurotransmitters as well. And another class of molecule that you should probably remember a little bit about from cell biology is the steroids. And steroids are neutral lipids, and all of these are going to be derived from cholesterol. And examples of these are going to be testosterone, estrogen, other things like these. And again, you should probably remember some specifics about these from cell biology. For instance, they have internal receptors. These receptors for these are not on the cell membranes, but internal in the cell, because steroids can pass very easily, being neutral lipids, very easily through cell membranes. But we'll come back and talk a little bit more about that in a second. Another kind of interesting thing about these hormones is that many can be very, very structurally similar to one another, but still have very different effects. So if you look over here on the left-hand side, this is testosterone. And on the right-hand side, what we have over here is estradiol, one of the major components of the thing that we call estrogen. And both of these are steroid hormones. They're also generally associated with gender-specific or sex-specific, functions. They're very, very similar. Again, they're both steroids, so they're very, very similar. However, they have, even though they act on kind of somewhat similar types of things, they have very profoundly different effects on the end result of what they have in the body. So, again, several different hormones can be structurally very, very similar, but still have very different effects. Another way that we can kind of classify hormones is by their chemical properties. So, a lot of these hormones can be, for instance, hydrophilic. I didn't spell hydro right there. Excuse me, there should be an R in there. Hydrophilic. And these, again, are going to easily be dissolved in water or aqueous solutions, and that would be the peptide and protein hormones, the catecholamines. And all of these, again, this is a kind of a harkening back to cell biology, all these should have receptors that are on the membrane. They can bind directly onto some sort of protein receptor on the membrane. However, we also have the lipophilic hormones. These are the steroids, and ones that we haven't exactly talked about quite yet, the thyroid hormones that we'll come back to later. All of these are going to have internal receptors in the cell. Again, these lipophilic hormones can pass very easily through the cell membrane, get into the internal cytosol of that cell, and they will bind onto receptors inside the cell. Okay, so that is how, that's kind of how we divide many of the major groups of proteins. Let's take a minute and start talking a little bit about how do we make proteins and why is this really important. And really that depends a lot on the class of hormone that we're talking about. So first off, the peptide hormones, this is really going to be similar to how most proteins are synthesized, and being, kind of in a biologically oriented major or pre-med or whatever you are, this should be a very familiar pathway to you. Essentially, we're going from DNA to RNA and then translating that into a protein. That is one of the fundamental dogmas of biology, and so that should be very familiar to you. So again, we're going to get some sort of translation by a ribosome, and this is going to become what we call the pre-pro hormone, the very first translation of the amino acid sequence is called the pre-pro hormone. And generally, after this initial translation, we're going to get some post-translational modification. Oftentimes, certain parts of these are going to be cleaved off, changed in some sort of way, and then this becomes the pro hormone, and then oftentimes with some additional post-translational modification can become the active hormone, or what's actually going to be able to dump into the circulatory system and moved off through wherever and have its effect. Some move directly from pre-pro hormone to active hormone. Some have this intermediate step, the pro hormone. But all this is happening by some sort of post-translational modification. And again, this isn't going to necessarily happen right away after translation. Sometimes this can happen quite a while after when we want to actually release it. And sometimes this post-translational modification can actually happen in the circulatory system. Not always, but sometimes. And what that means is occasionally, not always, but you can actually have prohormone circulating throughout your circulatory system, and it's just essentially waiting there for some sort of post-translational modification to become active and then actually carry out its function. And it gives you a little bit quicker way of activating some of these and actually getting these hormone signals out. Now, most of the time, these peptide hormones are going to be stored in vesicles packaged up by the Golgi and just waiting for release. And so they're just going to be kind of waiting there. And again, as I was talking about before, some are going to be further modified after their release. So there's going to be actually be inside the circulatory system. They're going to be modified at that point. Okay, so that's the peptide hormones. It's really kind of the same story that you learned about earlier in your classes, specifically about how proteins are expressed and made. It's really not that much different than that. But with steroids, we have a little bit different thing going on, and all steroids are going to be synthesized from cholesterol. And so this right down here in the lower right hand corner should look, well, first off, this is cholesterol. And this should look quite a bit similar to what we were seeing earlier with the testosterone and estrogen or estradiol. And all of these steroids have this characteristic 6-6-5 4-ring structure. So we have these 4 rings, and again we have number 6 ring, a 6 ring, a 6 ring, and then this 5 ring, 5-member rings all kind of bound together in this structure, and that's forming the basic kind of structural component of steroids. And the thing about steroids is, again, remember, like most lipophilic molecules, we cannot hold them very easily in some sort of bilipid membrane-bound vesicle or cell or something like this. And what this means, then, is that steroid hormones can't be stored after synthesis, and we can't just hold them there waiting to release them. They're going to start diffusing out of the cell as soon as we make them, because they easily diffuse through our membranes. And what that means then is that the rate of secretion of these steroid hormones is going to be set by the rate of production. As we produce them, they're going to be secreted pretty constantly after that happens. Now, this should also make a few other things go off in your heads. Okay, so these are lipophilic, they're diffusing out of these cells because they don't interact very much with those lipids there. But lipophilic things don't really dissolve very well in aqueous solution. So how are they being circulated by the blood? And I'd say, well, that's a very good question. Thank you for asking. It turns out that all these steroid hormones need to be bound to plasma proteins to be able to circulate through the circulatory system. And there's this sort of dynamic equilibrium. Not all of them are going to be bound to the plasma proteins. There's going to be this very small percentage that are free, but the vast majority are bound. And that's because we have this very, very low solubility of steroid hormones in the aqueous solution that is the blood. And so we're going to have this small amount that's free, and it turns out that only free steroids can bind to the receptor. If they're bound to the plasma proteins, these transport proteins, they can't bind to the receptors. They have to be free. So as they bind, more are going to pop off the transport proteins and come out. And then also the final class, again, the amines. Most of these class of hormones are going to be derived from tyrosine, and very much like the peptide proteins, peptide hormones, they're going to be stored in vesicles until released. We don't have the same problems that we have with lipophilic hormones. They can just be stored, released, kind of your standard run-the-mill fare there. Okay, so let's start thinking about the specific actions that each of these classes of hormones tends to have. Let's first look at these lipophilic hormones. Now, lipophilic hormones will often change gene expression. Now, if you remember, some of these that we're talking about were testosterone, estradiol, or essentially one component of estrogen. And you probably understand that the amount of either of these that you have during development plays a large role in the development of secondary sexual characteristics in essentially all animals. And they're doing that because they're changing gene expression in the organisms that they're being expressed in. And so that's kind of a characteristic of most of these lipophilic hormones, is that they're changing gene expression. And remember, they're binding to these internal receptors inside the cell, passing straight through the membranes. And then once this internal receptor is activated, it's going to bind to some element on the DNA. And on the DNA, what these are generally called are hormone response elements. And once this complex of the receptor plus the lipophilic hormone binds to that hormone response element, it'll tend to activate some gene that's associated with that element. Okay, so let's take a moment here and talk about how the endocrine system is regulated. We talked a little bit about this when we're talking about the nervous system and reflex loops and things like that. And we have something similar in the endocrine system, where essentially we start out with a sensory cell, and we're going to move on to some sort of integrator, something that's going to combine the sensory input of some factor, some environmental stimulus, and that integrator, in the case of the endocrine system, is going to be some gland, which is then going to release a signal, the hormones, going to be carried through the circulatory system and have some effect on some effector that is going to carry out some response. And in the case of the endocrine system, that effector is the target cell. Okay, now in the case of these integrators, the hormones could possibly be also stimulating the production of other hormones. So it's not necessarily that we have just a single gland that is responding to the sensory cell and on. We, in certain cases, can have some hormones that stimulate the production of other hormones. And just like in the nervous system, where this allows us to get some sort of integration of signals, essentially pulling multiple signals together, or get divergence of signals, that's doing the same thing here in the endocrine system. We can actually get integration or divergence of signals based on some hormones stimulating other hormones. And if you have a hormone that does, in its primary effect, stimulate the production of other hormones, that is what is known as a tropic hormone. So a tropic hormone stimulates the production of other hormones. Okay, the opposite of this is a non-tropic hormone, which is a hormone that isn't tropic. It doesn't stimulate the production of other hormones. Pretty straightforward. So as we're talking about how this is regulated, the primary factor that we're going to regulate with this is the blood plasma hormone concentration. This is really important. So again, remembering, just kind of hearkening back to the principal underlying factor about what the endocrine system is that essentially we are sending signals through the circulatory system to distant cells. And what that means then is that the amount of signal in the blood is like a very, very foundational factor of how these signals are going to be transmitted. So this is the amount of hormone in the plasma is a very key regulated aspect of the endocrine system, or in terms of how it's communicating. And there are several ways that we can do this. First off, we can regulate the rate of secretion. You increase the rate of secretion from a gland of some hormone, it's going to increase the amount of hormone in the plasma. Or we decrease the rate of secretion, it will decrease the plasma hormone concentration. Seems really straightforward. Okay. That's the one most people would probably think of right off the top of their head. But also we have a couple other ones. One of them could be the rate of metabolic activation. Now remember, just a few moments ago I was talking about how some hormones to really be active need to be modified, sometimes in the circulatory system, to become active. And so oftentimes the rate of this activation is one of the ways that we regulate the concentration of hormone in the circulatory system. We might have lots and lots and lots of some sort of prohormone circulating through the circulatory system, but none of it's actually being modified into the active form, and so actually we have very little of whatever hormone in the circulatory system. Now that can change. If we up the rate of metabolic activation, we can get lots of it all of a sudden. But this rate of metabolic activation is another primary way that we can control the plasma hormone concentration. Now, in specifically lipophilic hormones, we can change something else, and that is the binding rate by which this lipophilic hormone is going to be binding to the plasma protein. If it's binding at a very high rate, that's essentially taking it out of the circulatory system, because it's not going to be able to bind to the receptor, or vice versa in that case. Also, we have the rate at which the hormone is essentially broken down and removed from the circulatory system. So even if we're secreting a lot, if we're removing it very quickly, we're going to keep the level of plasma hormone very low. So the rate of metabolic inactivation of that hormone, or removing it from the circulatory system, is another way in which we can regulate the amount in the plasma. Okay. So with all that, there's a couple principal also regulatory mechanisms that are happening here. I'm going to just give you the names at the moment, but I'm going to expound more on these in just a moment. The first one is a negative feedback loop. Sometimes lots of hormone will inhibit more release of that same hormone. These are typical negative feedback loops that you expect to see in biology. However, remember I also said that the endocrine system and the nervous system are sometimes somewhat linked. We can also get neuroendocrine reflexes that also regulate the amount of hormone that's being, or the level of the concentration of hormone in the plasma. And then finally, also, occasionally we also have temporal rhythms that can set the amount of hormone circulating the plasma as well. Rhythms in time that will increase or decrease the amount of hormone. Now again, let's talk about each of these kind of in turn and expound a little bit more on that. So, just kind of to remind you what a negative feedback system is that the output of some system counteracts a change in the input. So essentially what we have going on here is that if some factor gets high, something gets too high, it will tend to cause a consequence of something that will lower that back down. Or if something gets too low, it will enact consequences that tend to raise that set point. So kind of the typical thing here is like a thermostat. Temperature gets too low, thermostat detects that, turns on the heater, raises the temperature. That's a negative feedback loop. Or temperature gets too high, turns off the heater, turns on the AC, and brings the temperature back down. Essentially, whatever factor, there is some sort of loop that pulls that back within a range of a set point. And many hormones are essentially part of a negative feedback loop in that these hormones will tend to inhibit their own production. This is a very, very common theme in hormone production. And again, as we look at very specific examples, you'll see this more and more, and just kind of leave it there at that for the moment. We also have these neuroendocrine reflexes as well. And I'm not going to talk a lot about these right now because there is quite a bit that we're going to talk about this later. But the specific thing to take away from this is that when we combine this with the nervous system, that we can get very sudden increases in hormone secretion in response to some sort of external stimuli or something like that, or an internal stimuli. And the key component of these is that these neuroendocrine reflex loops are going to contain both a neural and an endocrine component. So the nervous system is going to somehow be involved in this, and along with the endocrine system. And then we're going to kind of finish up this talk today by talking about these temporal rhythms in hormone production. And so, a lot of times, in some cases, what we have is hormone plasma concentration that will function or fluctuate as a function of time, going up at certain times, dropping at other times as well. And this is going to be partially regulated in the central nervous system. We have some sort of endogenous oscillators. And by endogenous oscillators, I just mean some sort of signal that oscillates up and down rhythmically. And these were commonly known as biological clocks. And endogenous oscillators mean something internal that is essentially varying in time, regularly varying in time. But we also have external cues that essentially set these biological clocks. And those are specifically called Zeitgebers. So Zeitgebers are some signal that is going to set the biological clock. Essentially, make sure that is coordinating in time. And there's various time periods that these sort of oscillators will operate over, or some time periods that these hormone concentrations will fluctuate over. We have, for instance, circadian rhythms, which I'm sure you've probably heard of before, and these are daily. We also have circadian, which tend to be monthly rhythms. And then we also have circannual rhythms, which are yearly. Now, circadian, or for instance, your sleeping, waking cycle, things like this. Circalunar, these tend to be more common in, for instance, most animals that have some sort of monthly rhythms, but then also there are some notable ones in humans as well that are monthly. And then finally, circannual, especially with animals that have some sort of, for instance, mating season or something like that, you have these rhythms that tend to be yearly in their period as well. So let's talk about how these, or one mechanism by which these can be kind of, give you a couple examples of these. So for instance, a couple different circadian rhythms that humans have come in the concentrations of melatonin, and this is one of the principal regulating hormones in humans and in many vertebrates, regulating a lot of different physiological functions. And these are melatonin, the production and secretion of melatonin is going to rise during periods of darkness and kind of setting this circadian rhythm. On the other hand, at periods of waking, we also have another hormone being produced called cortisol. And this kind of generates another set of physiological functions, often associated with alertness, waking, also can be produced during times of stress. Now, a lot of these, it's really interesting how your body is able to time the rising or the falling of the production and secretion of these hormones. And it primarily happens, the very core of this regulation of these periodic increases and decreases in production is going to happen in a place in the central nervous system in the brain called the suprachiasmatic nucleus. This is actually, suprachiasmatic is actually locating the space in which this happens. So this is in a part of the brain that sits kind of right above the joining or kind of the crossing of the two optic nerves. So it's fairly kind of far forward in the brain. And this really is the master biological clock in most vertebrates. This is setting the daily and sometimes even monthly or yearly rhythms in many, many organisms. And this is what controls the release of melatonin and cortisol. Okay, so let's take a few minutes to just talk about how in the world does this function? How in the world is this able to set this sort of regular rhythm? And first off, let's talk about how it gains the rhythm or gains kind of timing of the rhythm in the first place. It needs a Zeitgeber. Some external stimulus that's going to essentially set when this happens. And in the case of melatonin and cortisol, this tends to be light detection. So what you have is, in your retina, you have a set of specialized photoreceptors that actually are not detecting light that you consciously perceive. They're detecting light and passing it on specifically to one area that, or not just one area, it's transmitting its information back to the suprachiasmatic nucleus. This ends up being about 1 to 2% of all your photoreceptors are these kind of photoreceptors. And they don't just produce normal, or they don't just have your normal rhodopsin or scotopsins. Instead, what they have is this thing called melanopsin. And when this is going to be excited, it's going to, just like we were talking about in the sensory system, produce action potentials that go down the optic nerve, but it's going to send this light information directly to the suprachiasmatic nucleus, not off to the vision centers of your brain. So it's actually not helping you at all to produce an image. It is just helping you set the time. And so what this means, or essentially what this is that light in humans and most other vertebrates is the circadian zeitgeber. This is what is setting the timing of this daily signal. However, this circadian rhythm is going to produce in constant light or dark. If I took one of you and I put you in consistent dark for weeks, you will still rhythmically get tired, go to sleep, and rhythmically get back up again. Now the problem is you'll gradually get unsynced with the actual day, with the 24-hour day. You won't actually stay in sync with this. But what this means is not just light that's setting the time period. There's something internal. There's some mechanism that's still keeping that day, just like the mechanism of a clock. It just doesn't keep very good time. And so you need it to daily be reset by the action of these specialized photoreceptors that reset the clock every day and help it to get the right timing. But it continues. It doesn't need this light stimulus to actually keep time going or keep these oscillations of melanin and cortisol going. So how does this happen? Well, if we go back to the suprachiasmatic nucleus, there are a couple genes being expressed there, clock and BMAL1. And both of these are being expressed. And just like any other genes, they're going to be expressing some sort of protein that's associated with these. Well, these protein products from clock and BMAL1 are going to be transported back into the nucleus of the nerve cells in the suprachiasmatic nucleus. And what they do is they tend to activate, first off, themselves. So clock will activate clock, and clock will activate BMAL1, and BMAL1 will activate clock, and BMAL1 will also activate BMAL1. It'll activate itself. So this is actually not a negative feedback loop. This is a positive feedback loop where essentially something being expressed, specifically clock and BMAL1, and having these in the nucleus of the cell causes increases in their own expression. Kind of interesting. However, both of these, clock and BMAL1, will also increase the production of these two other genes, period and cryptochrome. Okay, so that's kind of interesting. So all of these, the expression of all four of these is going to increase. Okay, so all of these are increasing. However, there's a catch here. Period and cryptochrome then inhibit the expression of clock and BMAL1. And so as we get more period and more cryptochrome, we are going to decrease the expression of clock and BMAL, which means that we are no longer activating clock and BMAL, and so the expression of all four of these decrease. And all four of them are going to be broken down and removed from the cytosol, both the mRNAs and then also the proteins. So then we get a decrease in all four of these. And then at the low end, we're going to get period and cryptochrome. Okay, so after this, we get low levels of period and cryptochrome. But that means that we still have, there's still a little bit left. And there's also a little bit of clock and BMAL1, which then lead to increases in the expression of clock and BMAL1 and period and cryptochrome. So essentially what we get is something that looks like this. I'm going to kind of draw all over the front of this. If we get a graph, what we get is an increase in the production of all four of these. And that's because we got increasing clock and BMAL1. But then there gets to be a point in which there's so much period and cryptochrome that we're beginning to decrease the expression of all four of these, and that happens like this. And then when we get low, clock and BMAL1 take over again, and then we get increased expression again. And so it's this kind of cycle of large kind of increasing and decreasing expression of these 4 genes that end up, and there's a little bit more to this. These are then hooked up essentially, or coupled to the production of other hormones, but these will then, this rhythmic increase and decrease in the expression of these 4 genes can control the rhythmic expression of other hormones that then set physiological processes that are going all across your body. For instance, getting tired when it gets late, and then also, being wakeful and waking up. But here's another thing to keep in mind, is that this rhythmic cycle is being set by light. And so essentially what we're saying is when it gets light, we go into a certain phase of this. We essentially reset this. And this is kind of one of the depressing things, because I kind of like to watch TV late at night and stuff like that. That's really bad for this, because essentially we are resetting our schedule, resetting this expression. And so it's just something to keep in mind too. Well, there's a couple more ways that things can be regulated, how we can actually regulate the response of these hormones. And it's not just through how much protein, or the concentration of the hormone in the plasma that we've been talking about all the way up until here. We can also change a couple other things. And one of them that we can change is we can change the target sensitivity to the hormone. Essentially what we can do is up or down regulate the number of receptors that are on the target cells. And so another way that we can change the response is to make the target cells less sensitive or more sensitive to whatever hormone. In addition to this, we can also have hormone interactions. And this is another thing that happens fairly commonly as well. And there are several different ways in which they can interact. The first one is permissiveness. Now, in permissiveness, what this means is that one hormone must be present for another hormone to have an effect or a maximal effect. If you take that hormone away, essentially this other hormone will not work. So again, let's just kind of, for hormone A to work, imagine, B also has present. It has to be present. So A plus B, we get an effect. If we just have A, no effect, or reduced effect, and just B, no effect either. So this is permissiveness. We also have synergism, where essentially 2 hormones act together to amplify an effect. So if we have A, we get small effect. If we have B, we have small effect. But if we have A plus B, we get large effect. Now remember, this is one way to look at this as being different than permissiveness, is that, for instance, remember A is the actual effector hormone, but B just needs to be there for it to work. But B doesn't have any effect on its own. That's permissiveness. Synergism, where they each have their own individual effects, and together they essentially amplify their effects. And then also we have antagonism. Antagonism specifically is where one hormone is going to cause the loss of function of another hormone's receptors. And so this can either be changing their function or removing their function, or actually removing the receptors themselves. So some hormone actually causes those receptors to go away. And this is antagonism. Okay, so all of that should really give you a good kind of foundation background of what's going on in the endocrine system and the basic principles by which this is working. Tomorrow, or the day after tomorrow, the next thing that we're going to be going into is very specific systems and very specific sets of hormones to kind of flesh this out a little bit more. I talked a lot very much this time in generalities and what basically happens without giving you a lot of very specific hormones. That's where we're going next. We're going to start talking about and putting all these principles into action, seeing how this actually functions with actual sets of hormones. Okay, talk to you later.